All, this is Dr. Mubin Sayed from drbeen.com. Welcome to one more show. So this is a super secret chit chat about the study and your questions. So let's see if we can talk about that very quickly. This is how Rumble is. I actually do not myself know how to use it, but this is what the Rumble is. What I do not know is that once I go live on it, what how will you find it? Is there a notification, or do I send a link? Do I tweet it? Do I put that in YouTube have a policy, and I think rightfully so, that if you put links to another platform, they have a right to remove your channel. Although I've seen many people doing it, but at least they have that policy. So if they ever want to hit me, they can do that. But we got to figure this out, how this would work. We'll do it tomorrow then. Now, let's see the questions. Alquin says Rumble's is 5 times less cute than YouTube. It seems so. Was Odyssey better? I have to. I think there is one way for me to do this Odyssey with correct chatting experience as well. If I use OBS and connect Odyssey to OBS, I believe OBS can pull the comment stream, and then you can display the comment as I do that here right now. So I'll have to learn OBS. Look how many things I have to learn. Texas Meg says, and we still don't know about turtles and Rumble. Is Margaret here? Okay, so DDS is back here with zombies. Carolyn says, found you. Thank you. Sita says, hello again. Bambi is here. Hello, Bambi. How are you? Okay, so Margaret says, I'm here. Excellent. Quinn says, Barbara says, I just signed up for Rumble. It wasn't easy because it said another app was drawing over other apps, so I couldn't install Rumble. Facebook, I'd signed up for a course. Thank you. Keeney says, download Rumble. I'm so grateful. John says, you can do YouTube on Rumble and Rumble at the same time. Yes, the problem is there are some things I don't want to do on YouTube, which YouTube does not like. So that's why I'm trying to figure out if I can go to Rumble or Odyssey or some other platform. Dr. Z is here today. Hey, Dr. Z. So this is a good question. So I'm going to now thank you for getting us started, Dr. Z. So the question, what can we extrapolate for us? It's actually, this study is less scary if you think about adenovirus-based vaccines creating intentionally by design DNA fragments in our nucleus. Remember, for an adenovirus-based vaccine, we had done this discussion before, that what happens is, let's say here is a cell, and this is a nucleus, and nucleus has nucleopores, holes in it. These are you can say, transport gates. So when the adenovirus arrives in this cell, it is in a phagosome, it is in a small structure, it is captured, right? And then what we do is we have lysosomes, which are stomachs of the cell. We fuse the lysosome and phagosome and spill the acids on the virus, and we try to break it down. So when the virus comes out of this thing all beaten up and shattered, we only have the virus's shell, half broken, half intact, which still is carrying the DNA material for spike protein. Remember, adenovirus-based vaccines have a complementary DNA, not RNA. This adenovirus carcass is pulled by machines and attached to our cell's nucleus's nucleopore or hole. From here, this virus would then allow the DNA to go in the nucleus. Then, inside the nucleus, this little gene will become messenger RNA that would come out of the nucleus hole, then go to the cytoplasm, work with the ribosome, make spike proteins, and then the rest is history. So, this vaccine actually produces DNA fragments in the nucleus, and we are not worried about that. And here we are saying that there is a possibility of some DNA fragment by messenger RNA vaccine produced, let's say, outside or inside, we do not even know yet. So, one, I'm not worried because of that. Second, if this study has any merit to it, this study, and if their observations have merit, then what they're finding is that in the nucleus, even if it is integrated, this is the genome, even if this piece of DNA is integrated here, it is integrated in incorrect fashion from 3'. And because of that, it cannot be transcribed. So it's kind of a garbage gene added without any function, because machines that need to make a copy of this, they cannot engage with it. Imagine you have a photocopying machine and you have a book or a page which is just totally not going to fit on that photocopy machine. Then you will not make photocopy from that. So it is not going to make a photocopy. That means nothing would happen. That is one. I still doubt the idea that the genome will actually integrate the SARS-CoV-2 vaccine gene. If that was happening, then we have given billions of vaccine doses, we would see an expression of spike protein all the time. Now, there are studies that are saying that the spike protein may be expressed for a long time. I have to look at that study. That is one of the study that is in my mind to review. So going back to your question, if we extrapolate it, let's look at the scary parts or the good parts. Scary parts are going to be this gene gets integrated and is in the correct order. That means it makes spike protein. If that is the case, our immune system is not going to forgive these cells and going to attack them and kill them. And that is what these researchers say as well, that in some cases there has been hepatitis, for example. There has been case reports on individuals who developed autoimmune hepatitis after BNT162b2 vaccination. And they are saying that what happened was that the cell expressed these spike protein pieces or spike protein, and then the immune system that was already, let's say, from one dose or previous experience, exposure to the virus, knew to attack such cells, it came and started killing it. I actually think that autoantibodies can be produced in general. So in some people, autoimmune diseases can occur from vaccines or the virus. We are seeing them. So scary parts, it is viable gene, it makes spike protein, then ends up causing damage to the cell. That's one. Second, scary part. So scary parts first, and I've already said why I'm not worried. Second scary part, that the gene sits down in the nucleus, causes genotoxicity and causes the whole cell and the nucleus to go under attack or malfunction. Two, thirdly, are dividing cells, for example, immune system cells or the GIT cells or the surface cells, they become malfunctioning or develop cancers. Fourth, if the vaccine or this DNA vaccine goes through placenta to the baby and the baby's dividing cells make this DNA, then what is the effect of baby? We don't know. Although now from vaccines, we know that this is probably not something that is observed, but if we extrapolate that what can happen, these are the unknowns. So immune system responses, genotoxicity, carcinogenicity, child's uh, birth defects maybe, but have not really been seen. And as I said, I am less worried because um, adenovirus, if these are the things to worry about, then the adenovirus based vaccines are more scary because they directly make genes in the nucleus. The question that is in my mind, and I haven't yet been able to answer that, is how long this DNA stays. So when the researchers would start fighting or debating where this DNA is, I guess then the next step will be to see what is the outcome of this DNA and then to see how long it sits in there. That discussion is still not happening. Joy Fisher says, I'm leaving this chat to watch the prior video, which I missed. Cool. Doug is here. Hey, Doug, how are you? Alicia says, hello from Palm Springs. Hello back to you, Alicia. Nick says, was there a reasonable Pfizer LNP levels in the assay compared to the level seen in the mouse model, biodistribution model? So they tried to create levels similar to the vaccine in the muscle. So they actually have a section in their study where they say, here, fortunately, we are right on that section. So if you see here, they say, therefore, in this study, or I'll have to read this whole paragraph is about the dosage. Intracellular accumulation of lipid nanoparticle in hepatocytes has been demonstrated in vivo. A preclinical study on BNT162b2 showed that BNT162b2 enters a human cell in human EK293 cells and leads to robust expression of BioNTech antigen. Therefore, in this study, we first investigated the entry of this vaccine in the liver cells, hepatoma cells, the choice of vaccine concentration used. So, this is the answer to your question, Nick. The choice of BNT162b2 concentration used in the study warrants explanation. BNT162b2 is administered as a series of 2 doses 3 weeks apart, and each dose contains 30 microgram of BNT162b2 in a volume of 0.3 milliliter, which makes the local concentration at the injection site at the highest 100 microgram per milliliter. A previous study on the mRNA vaccine against H10N8 and H7N9 influenza viruses using a similar LNP delivery system showed that the mRNA vaccine can distribute rather non-specifically to several organs such as liver, spleen, heart, kidney, lung, and brain. And the concentration in the liver is roughly 100 times lower than that of the intramuscular injection site. So they said, we know this much from various studies that in the muscle it is 30 microgram per or 100 microgram per milliliter. Then from other vaccines, we know that the liver gets 100 times lesser. So they're going to try to produce a similar dose. In the assessment report on BioNTech provided to EMA by Pfizer, the pharmacokinetic distribution studies in rats demonstrated that a relatively larger proportion, up to 18% total dose, was distributed to the liver. We therefore chose to use 0.5, 1, and 2 microgram per milliliter. So remember, in the muscle, 100 microgram per milliliter. Here they went for almost 100 times less, 0.5, 1, and 2 of vaccine in our experiments on the liver cells. So that's what they did. So Nafthiel says genotoxicity section, he will look when he can, I'm sure. So we already looked at genotoxicity. We discussed it last time as well. So here, genotoxicity. In the BioNTech vaccine toxicity report, no genotoxicity or carcinogenicity studies have been provided. Then they say over here that And this may give rise to the concern if BioNTech derived DNA may be integrated into the host genome and affect the integrity of genomic DNA, which may potentially mediate genotoxic side effects. Which genotoxic really means that our DNA becomes unstable and our cell just destroys it to saying, what the fuck? What are you doing? And why do you have this extra gene? And it would just attack the DNA and try to repair it. And if it cannot repair it, the cell would do apoptosis and die. Julie says, Dr. Bean, thanks so much for what you do. Love your lecture. Thank you very much. Doug, Doug is laughing. Roderick says, so it can kill you. What? Did I say that, Roderick? They said we do not know about the genotoxicity. I extrapolated or explained what genotoxicity can mean. Doug says, WTF. I sometimes it just slips. Sorry, Saraswati. Yes, I said. Helena B says, are you getting enough sleep? Better than before. So if you can see, my eyes usually become a little, little tense by this time. I am waking up for last 2 days at 3 or 4 again, but this time it's not Luffy. I think it is I'm trying to sleep earlier. So my sleep became disturbed a couple of times, but generally I'm not in that state of mind as Luffy had done. Ray says, where is your cat? I am sure he's at this time outside. Augie says, hit the like. I think we should do a session with Luffy. I should keep him here one day and then just have him. Okay, so let's keep answering those questions. Um, Denise says, have you seen Dr. Osgood's latest tweet about seeing barely any inflammatory cytokines with Omicron long haulers and instead highly elevated markers of platelet activation and angiogenesis? No, I did not, but it is interesting. Microclotting could be an issue. I really see this tinnitus with Omicron is a very clear example for me. Now I'm talking Dr. Mobeen, giving my opinion, medical opinion, not giving to anyone. I'm just thinking aloud. This is not a study. To me, the tinnitus is an example of the microclotting causing blood supply disturbance and damage to the internal ear hair cells. So Mama Knows Best says, so this means our cells can identify themselves as not right and essentially kill themselves off in a good way? Yes, absolutely. So our cells, I wish we can learn from our cells how to live our life. Uh, our cells, when they're dividing, so let's say this is a nucleus. When they're dividing, remember there are various phases of the cell, mitosis and meiosis and so on. In these phases, when the new DNA, because we're going to divide the cell into two daughter cells, there is one copy of the DNA. We want to make one more copy. When the DNA copies are made, the cell halts in that state for hours and days, and our enzymes go over every single piece of our gene that is duplicated and confirm that it is correct. And if it is not correct, they actually repair it. They don't just note it down and say, all right, DNA bad, go away, let it be a cancer cell. No, they actually repair it. And there are so many kinds of repairs. It's fascinating. Sometimes, as you can see, I like mechanisms. So I can visualize myself sitting in there in one of those little tiny cranes that is going over the DNA road and finding a pothole and then fixing it. So they can excise off the damaged parts and they can then repair them with the correct parts. And if they are unable to do it, then they kill the whole cell. They say this cell is no good, it is not allowed to continue anymore, and they kill it. A new cell will be formed, but this cell is not allowed to be there. Similarly, if a developed cell, stable cell, develops the DNA damage, then our cells try to fix that as well. And if they cannot fix it, they tell the cell to die or cause apoptosis. So then your question will be, then how come there are cancers? Because cancers are example of DNA issues which are not repaired. So yes, sometimes that repair fails, and that causes cancers. Very sophisticated machine our body is. I spoke like Yoda. Very sophisticated machine our body is. So Raghav Patel says, do the vaccines have room for improvement? Yes. So I think the design, that what epitopes, then the dose, and then the type of what is that, the adjuvants, that how much of the immune system botheration they create and what kind of side effects they can create, that all can be looked at. PS says, can you please talk about XOCT24 and aviptadil? Okay, I will. Roller Girl says, hi, Dr. Bean and everyone. Hello, back to you. YouTube user says, Mother John's really slammed Bruce Patterson. Did you get pushback from it too for interviewing him? So I think maybe majority of the reason I think I got other kind of issues. My issues are mostly people just going to agencies and complaining about it, and then agencies doing stuff. Um, I think because I had, and this is true, I had my full disclosure. I said that there is no interest I have in this whole thing. I am more interested in the mechanisms. So I think because of that, there was some leeway given, but maybe at some point there will be some attack on me from that kind of a sector as well. But a few days ago, the attack which really made me surprised was from a friend of ours, and he attacked me so badly, and that caused his followers to attack me in also in such just mean and horrible way that I looked at those messages and I said, I this is what I've always thought, that there are no humans like that can do these or say these things, but they were. So I am attacked in a different way. Equal opportunity attacks are occurring. So John Snyder says, just looked it up. 2.1% of SARS-CoV-2 ended up with hepatitis, and a large portion had liver disease. Yeah. Hepatitis during the viral infections or general stress on the body can occur because there is a big demand from the liver to, one, do the energy metabolism, secondly, have the inflammatory system produced, and then have the onslaught of all the pathogens and all that. So liver becomes um, a target in some of these cases. Plus, it has to work harder. So it can be a transient hepatitis. Sometimes it is not transient. Sometimes there is fulminant liver disease and the person dies. You mentioned tinnitus from Omicron. Could it be possible that vaccination could cause tinnitus in the same way? Yes, it is possible. Yes. Vince says, if you could go back in time, would you still get the mRNA vaccine? So, Vince, is that a setup to get me yelled at? Because I'll say yes, and then people would say, you are just crazy. I like mRNA vaccines. And if you think about it, compare it to the adenovirus based vaccine. That is the second choice. So the choices are no vaccine, adenovirus based vaccines, inactivated virus vaccine, sub subprotein vaccine, and mRNA, right? So let's see. No vaccine is one choice. And whoever has made that choice, that's their choice. don't have any good or bad to say there. Then let's say inactivated virus, then adenovirus. And within the adenovirus, there are then two type. One are homologous adenovirus doses, that is Johnson & Johnson and AstraZeneca, and then is the heterologous or heterologous, that is Russian Sputnik. Their first adenovirus is human type, and the second adenovirus is from the monkeys. That is the adenovirus. And then is the messenger RNA vaccine, and then is the subprotein vaccine. And now you're seeing Medicago like things. So delivery and production, adjuvants, there are changes in those formats as well. Generally this. So you could then say, for example, nasal or oral vaccine, and their delivery mechanism is different and so on, but they're still antigenic in one of these ways. So here, there are some folks who have this choice. And inactivated virus, we just don't have it here. I actually liked inactivated virus. The thing, if you go back to my videos, you would see that I would always just say that I get scared if the inactivation process is not correct for SARS-CoV-2, it can be deadly. But inactivated virus vaccines have been used a lot on the eastern side. Adenovirus-based vaccines. These are the vaccines that actually produce DNA fragments in the nucleus. So if we have to be scared of some vaccine, and this is a traditional platform, that means this kind of vaccine has been out there for a long time. Then is the messenger RNA, and then subprotein. And if you watch my videos, I always used to say that I would love to have Novavax, but Novavax is just not there. It's not there even now. And then in Moderna, in messenger RNA, I loved Moderna. So I think the basic question probably would be, will you still get vaccinated? So yes. And then based on what choices do I have, I might decide which one. Uh, Guardian Angel says, will Novavax travel to heart or through blood-brain barrier? Will it stay at the injection point only? So in theory, it should not go to blood or brain. It should be picked up by the immune cell, not even by regular cells. So regular cells will not become a target for the immune system to be killed. So it should only be picked up by immune cells. Because it is an antigen, it cannot go into regular cells through a lipid nanoparticle, so it just has to sit outside. And then immune cells are the one that are going to pick it up and eat it and destroy it and present it. So from all those mechanisms, it actually looks good. I have still to look at the clotting and myocarditis and those side effects. Novavax, Corbevax, Covaxin, yes. So meanwhile, in America says, how do you hypothesize long COVID triggers vertigo? Can it just make everything generally swell up in that area? Vertigo began after second dose, but other long hauler symptoms mostly felt better after for some time. So the vertigo has three main reasons. And vertigo can be temporary or permanent depending upon the kind of damage. But three main reasons. One is issue with the eyesight, because our body has three ways to balance, to understand the balance. And when the balance is out, that is when we feel the vertigos. One is the eyesight, that helps us to understand what is our orientation and balance. The other one is ear and the ear canals, that tells us our balance when we move our body. And the third one is the muscle and the muscle spindles and the Golgi tendon organs, the muscle tones and the tendons, they report our position. any one of them or two of them. Our brain needs at least two of them to work, to not create vertigos, but to say you are stable. If two of them are not working correctly, then brain figures out there is something wrong and we are probably poisoned, and then it creates vertigo. That is one way. Then there can be electrolytes, and there are other issues too, which eventually that is what brain is upset about, that maybe there is electrolyte imbalance or poison. So if there is an internal ear damage or inflammation, if there is inflammation, that is a temporary vertigo, and it will come and go as inflammation fluctuates. But as inflammation subsides, it can become okay. And the neurological system's inflammation in terms of SARS-CoV-2 I'm seeing can take 5-6 months. So it can become permanent if the damage to the nervous system tissue has occurred, or it can be temporary. And it really, I cannot make a conjecture unless it's looked at by through the labs and general physical and the clinical experience. And I'm sorry to hear this. No, no, that is not there. What is the take home message? Will I get cancer? No. So that is what adenovirus based vaccines make DNA fragments in the nucleus. So I'm just thinking about the cancer. So here is how cancers develop. So I, of course, cannot guarantee anyone or even myself that something will not cause cancer. Even light can cause cancer. Um, so here, just simple irritation of the tissue cells that causes cell damage and then needs repair can cause cancer. So here is a cell, and let's say the DNA got damaged, and the cell tried to fix it, and it could not fix. But at the same time, it could not regulate itself, and it could not um, do apoptosis. At the same time, the immune system, for example, natural killer cells or macrophages or the other toll-like or pattern recognition cells, they could not identify there's a problem. Normally, natural killer cells would find it out right away and kill the cell. So even they missed it. So number one, a change occurred which could not be repaired by the cell itself. One failure. Second failure, cell, if it could not repair something, it would actually cause apoptosis or die. That didn't happen. Third, when the cellular malfunction started occurring, our natural killer cell or immune system forgot or could not handle it as well. Third failure, then the cell could, and then the fourth failure, I mean, cancer has such a complex pathology. Fourth failure is cell went out of control of regulations. It just said, I'm not going to listen to anyone anymore. I'm rogue. So when all those failures occur, then the cell starts becoming duplicated and increased and increased. And we don't know the cancer until a specific amount of tissue has built up. And that can take time. So, for example, God forbid, I can develop a cellular malfunction today and still not know that I have cancer for months because the cell number has not increased enough to cause a problem, for which then I'll go to the doctor, and then doctor would do the tests if they could catch it in time and say, okay, this is cancer. Sometimes even doctors will miss it and they'll suspect not a huge problem, and then later on it turns out to be a cancer. So that is the only long-term type of outcome that starts immediately but would appear later. So, to answer your question, again, there is no guarantee, but at least this mechanism itself, if I stay with the study, they stop short of all of that. To me, it seems like either they didn't have the resources, or this is what they didn't want to discuss, or they knew that this is a landmine, so we just going to stay back and we just not touching that area. That is it. Uh, genotoxic or is it carcinogenic or not. They just didn't go there. So they stopped there. So if I just stay honest to the study, I would just stop there and say, don't know. If we look at other vaccines, their behavior actually is more aggressive than this one, adenovirus based vaccine. So Evelyn Spaghetti says, is this why I have had inflammation since the vaccine? So the inflammation since the vaccine or the virus is a long haul, and we have been discussing so many various mechanisms about long haul. Please, when you get a chance, watch some of my videos. I was actually trying to see if I can create a separate playlist to separate out the long haul so people can just see them in one go. I'll try to do that. But there are, if you just search in my channel for long haul, you'll see many studies. Ashita says, I want to do PhD in the user. Thank you. Um, Aether says, other than the spike protein, what other components of a SARS-CoV-2 virion is pathogenic? N protein is pathogenic as well. Of course, the whole virus is pathogenic because pathogenicity is not just the virus binding and getting into the cell, but just a foreign material in our body makes our immune system mad. So any part of it is going to be upsetting to us, our immune system. Leo says, I think it's been long enough. What test can we do to see what possibly relieve us from suffering? Aqua. Are you talking about the um, long haul? Please, there are so many long haul discussions that I've done. You can also do research on long haul COVID or vaccine, post vaccine injury, and there are many studies to look at. I think that there is a decent discussion in my Discord as well. Although it turns out Discord has just recently updated their medical misinformation, and so I don't know what they are going to start doing. This world is just all over the place. So Nekar Rus Nekar Sulmi says, yesterday's conspiracy theories are today's facts. I don't know which conspiracy theory. If you're talking about the DNA fragments, then I've been saying it again and again. DNA fragments are produced by adenovirus as well. The question is, what is the outcome of that? So, MHI have discussed NADs, NAC, and others. There are benefits of these things. None of them in one place and as one is sufficient to control something, but a balanced diet, balanced approach is important. Sherry says, can you do a talk about vertigo and dizziness? Yes. Yes. Deanna says, sorry, just hopped on. I didn't watch all, but I very much value your work. Extremely great. Thank you very much, Deanna. FLCCC has calmed the vertigo, so thank you. I'm very happy about that. Denise says, I've been using sanitized nasal spray for the past few months until my tier is being given prophylaxis monoclonal antibodies, being unable to have been vaccinated due to my health history. Got it. I like sanitize. Michelle, I'm so sorry. Positional vertigo. I know it is so difficult. My sincere sorry. Robin says, Night Paul Burke. Night Paul Burke. Doug says, Good night. Good night. Oh, I didn't notice it's already. Wait. Hi, Dr. Bean. Did you see that CDC now recommends longer interval for vaccination? I didn't, but it makes sense, and they should have learned from Britain. Britain had increased the interval and actually did it very successfully. The Ragu Overlord says, new to the channel. Welcome, Overlord, Mr. Overlord. Uh, Carlos Ortiz says, Hi, Dr. Bean. Did you see that CDC now recommend longer interval? Yes. This DNA liver influence concerns me. Waterhouse says, Dr. Bean, didn't you get the messenger? Yes, I got Moderna. DNA in liver cells. So this, look, let's use that study as information. What they're saying is, we can see it can go to liver. A lipid nanoparticle based vaccine can go into any cell because all cells are made up of phospholipid bilayer, and lipid nanoparticles function is to cross that. So they just decided to do it on liver because they said we saw there were people with hepatitis or the animals with hepatitis, and animal studies showing liver cell issues. So that's why they did it on liver. They could do it on any system which had the dividing capability, and they would see these kind of behaviors. So if you're worried about no. Robin, so that is a difficult question. What would be the treatment for autoimmune hepatitis or COVID or the vax? Would the liver damage be permanent? So liver is a very resilient organ. Normally it regenerates, even if it is very small amount left, it still works. Liver's basic problem occurs when the basal membrane of the liver starts getting destroyed, number one, and number two, the scar tissue causes so much of contraction that the blood supply and the function both starts getting affected. So generally liver can, that is why cirrhosis and cirrhosis damage takes decades. Now, your question, what would be the treatment for autoimmune? So, of course, there are some drugs that help liver, that support liver in general, but ideally control the autoimmune disease. So, there is a complete specialty for rheumatic diseases or autoimmune diseases. So that would be. In the past, we used to use steroids more often. Nowadays, we have more targeted antibodies and other drugs. Would the liver damage be permanent? That depends upon what is the patient's condition, what is the liver's condition, how much the insult occurred, how fast, how extensive, if the basal membrane got damaged. There are so many factors. Liver generally is much more resilient than other tissues. Cool. So with this, let's stop for now. It's 8 o'clock. I will be, when this gets on Rumble, I would delete it from here, and then we'll figure out how to continue to do our chit chats on Rumble. So thank you very much. There is a link in this description which is for access to Dr. Bean's premium content. And the link has a price that is the I hate to call it cheap because it is really low price, and my content, each video is more pricey than that. I love our content. So if you like, please buy a plan. And then in addition to that, there are links in the description. You can buy me a coffee, or you can be a patron, or you can use PayPal. And if you can, please like and subscribe and share. And I would see you tomorrow. Bye bye for now.